Welcome to another deep dive. Today, we're plunging into this uh, incredible, often unnoticed world inside basically every living creature.
That relentless pump, right? The silent flow keeping everything going.
Exactly. We're talking animal circulation. And our mission today is to really unpack the ingenious ways animals move blood around.
Looking at the core concepts, the different systems, why they're different.
Yeah, the adaptive significance. We're drawing heavily from a chapter in Animal Physiology by Hill, Wise, and Anderson. We use, real examples. Look at how scientists figure this stuff out.
It's fundamental stuff. And whether you're, say, an athlete pushing your limits or just curious about biology,
Right.
understanding circulation helps you appreciate these hidden lifelines. It's
Mhm.
way more than just a pump. It's this whole internal communications supply network constantly adapting.
Okay, so if it's about moving vital stuff around, why can't simple diffusion just do the job? Why do bigger animals even need this whole system?
That's a really critical point. The fusion works fine for tiny things, under a millimeter or so. Molecules just move down their concentration gradient.
Simple enough?
But for anything bigger, it's just hopelessly slow. Imagine trying to get oxygen from your skin to your big toe by diffusion. It'd take, well, ages.
Years, probably.
Exactly. So animals over that rough size limit absolutely require circulation. It's this rapid, pressure-driven bulk flow. Think of it like an internal express delivery service.
It's pretty amazing how the whole idea of circulation evolved, too, isn't it? Like
Yeah.
William Harvey back in 1628, he figured out the round trip.
But had no clue about capillaries or oxygen or hormones.
Right.
It shows how science builds, His idea was revolutionary then. But over time, discovering capillaries, then oxygen, hormones, immune cells, our understanding just got richer and richer.
It's this complex, vital network now.
Absolutely underpins everything.
Okay, let's get to the core.
The heart.
This discrete, localized pump. But I've heard some animals don't even have one. How does that work?
It's true. Some worms, for example, annelids, they just use rhythmic contractions of their blood vessels themselves, like a peristaltic wave pushing blood along.
Huh. No central pump.
But hearts are definitely the common solution, and they come in all sorts of shapes and sizes. Single-chambered in arthropods, multi-chambered like ours in vertebrates.
And even extra helper hearts sometimes. Accessory hearts.
Yeah, auxiliary hearts boost flow in specific areas.
So the heart muscle itself, the myocardium, it's got some special features, especially in vertebrates. What makes it unique?
Vertebrate cardiac muscle is really distinct. It is striated, like skeletal muscle, but the cells are joined end-to-end by these specialized things called intercalated discs.
Okay, intercalated discs, what do they do?
They're super strong mechanical links, but crucially, they contain gap junctions. These are like tiny tunnels allowing electrical signals to pass directly and very quickly from cell to cell.
Ah, so the signal spreads fast.
Incredibly fast. It means the whole chamber can contract almost as one unit, synchronously, which is vital for efficient pumping.
Got it. The whole chamber squeezes together. And the electrical signals themselves, the action potentials, they're different too. Longer.
Much longer than in nerves, hundreds of milliseconds sometimes. This ensures a good, strong, prolonged contraction to really eject the blood, not just a quick twitch.
Makes sense.
And critically, specialized pacemaker cells within the heart muscle itself spontaneously fire off these signals, setting the rhythm, like the SA node in us.
Okay, the SA node. Let's use the human heart as our main example then. That
Yeah.
classic four-chamber structure. Walk us through the flow.
Right. So think of it as two pumps side by side. The left side gets that fresh, oxygenated blood coming back from the lungs.
Into the left atrium, then ventricle?
Yep. And then whoosh, out the big systemic aorta to the rest of the body. Meanwhile, the right side is getting the used deoxygenated blood back from the body
Atrium, right ventricle.
and pumps that off to the lungs via the pulmonary artery to get oxygenated again. And you've got these passive valves, one-way doors, basically making sure blood doesn't flow backwards.
It's a neat system. So when we talk pumping mechanics,
Right.
contraction is systole, relaxation is diastole. How does that cycle work, say, in the left ventricle?
Okay, so during systole, the ventricle squeezes. First, both the inlet and outlet valves are shut. Pressure skyrockets inside. That's isovolumetric contraction.
Building pressure.
Then, bang! Once pressure inside is higher than the aortas, the aortic valve opens, blood gushes out, ventricular ejection.
Okay.
Then, as it starts relaxing, diastole begins. The aortic valve snaps shut, Both valves are closed again for a moment. Pressure drops, isovolumetric relaxation.
Then filling.
Then the valve from the atrium opens and blood rushes in to fill the ventricle, mostly passively at first. And the key measure of performance for any heart, really, is cardiac output.
Which is?
Just heart rate beats per minute times stroke volume, the amount pumped per beat. Total volume moved per minute. That's the bottom line.
Okay. Now, thinking about how hard that heart muscle works constantly, how does it get its own oxygen? It must be super demanding.
Oh, incredibly. It's one of the most oxygen-hungry tissues, right up there with the brain in vertebrates. And animals have basically evolved 3 main ways to feed the heart muscle itself.
3 strategies.
Yeah. In mammals and birds, like us, the myocardium is very dense, very compact. It can't get enough oxygen just from the blood flowing through the chambers.
So it needs its own supply line.
Exactly. It relies on a dedicated coronary circulation. Arteries branch right off the aorta, deliver oxygenated blood deep into the muscle, and coronary veins collect the used blood.
Which is why coronary artery blockages are so serious for us.
Precisely. Cuts off the heart's own oxygen supply.
Okay, that's the compact strategy. What are the others?
Well, in lots of fish, amphibians, non-avian reptiles, their myocardium is more spongy. It's full of open spaces like a sponge.
Ah, so the blood inside the chambers can reach the cells.
Right. The muscle cells get oxygen directly from the blood flowing through the heart chambers. The catch is that luminal blood, especially coming back from the body, isn't always super oxygenated.
Interesting trade-off. And the third way?
Is a mix. Mixed myocardium. You see this in active fish like tunas, some sharks, some amphibians, and reptiles. They have an outer compact layer with coronary vessels and an inner spongy layer getting oxygen from the chamber blood.
Best of both worlds, maybe.
Kind of. And what's really neat is the plasticity. Sockeye salmon. Different populations face different migration challenges. The ones with tougher migrations, they actually develop a significantly thicker compact layer. Demand shapes structure.
Wow, that's a fantastic example of adaptation. So what actually triggers the beat? Is it always the muscle itself, or can nerves be in charge?
Great question. This leads to the difference between myogenic and neurogenic hearts.
Myogenic means?
Muscle-generated, like ours. Vertebrate hearts are myogenic. The impulse starts within the specialized pacemaker muscle cells we talked about. Even if you cut all the nerves, a vertebrate heart keeps beating on its own.
It's intrinsic to the muscle.
Exactly. And in mammals, there's this amazing conducting system, specialized pathways that coordinate the signal spread. Ensures a slight delay so the atria contract first, fill the ventricles.
Then a rapid, coordinated squeeze of the ventricles.
Perfectly timed for maximum efficiency.
Okay, so that's myogenic. What about neurogenic? Nerve-generated.
Right. Here, the rhythm originates in neurons. The classic example is the lobster heart. It has this tiny cluster of neurons, the cardiac ganglion, sitting right on it.
Just 9 neurons, I read.
Yeah, tiny. Those neurons fire off rhythmic bursts, telling the heart muscle when to contract. If you take that ganglion away, the lobster heart just stops, completely reliant on the neurons. You find this in other crustaceans, horseshoe crabs, spiders too.
Fascinating difference. And we can actually eavesdrop on this electrical activity, right? With an ECG.
We can. That's the electrocardiogram. As different parts of the heart muscle depolarize and repolarize, they create tiny electrical currents that spread through the body fluids to the surface.
And the ECG picks those up.
Yeah. It gives us that characteristic waveform. The P wave for atrial activity, the big QRS complex for ventricular contraction, and the T wave for ventricular relaxation or repolarization. It's a powerful diagnostic tool, lets us see the heart's electrical health non-invasively.
Okay, so the heart has its internal rhythm.
Yep.
But it's not just fixed, right? Hormones, nerves, they can change the pace.
Oh, absolutely. It's constantly being modulated.
Yeah.
Hormones like epinephrine, adrenaline, they ramp up heart rate and force when you're stressed or exercising.
Fight or flight.
Exactly. And most hearts, myogenic or neurogenic, have nerve inputs too.
Mm-hmm.
In mammals, you've got the sympathetic system speeding things up and the parasympathetic slowing things down.
Fine-tuning the beat.
Yep. Even in the lobster, its cardiac ganglion gets signals from other nerves that can speed up or slow down the rhythm and intensity.
And beyond these outside influences, does the heart have any, internal wisdom, ways to adjust on its own?
It does. And a really crucial one in vertebrates is the Frank-Starling mechanism.
Frank-Starling, what's that?
Basically, the more blood that flows into the heart during filling, the more the heart muscle gets stretched.
Okay.
And the more it's stretched, the harder it contracts on the very next beat. It intrinsically matches its output to its input. More blood in, more blood out, prevents blood from backing up.
That's clever. It adjusts automatically.
It's fundamental for stable function. And interestingly, lobsters show something similar, but it seems to be mediated by their cardiac ganglion adjusting its output based on stretch rather than the muscle directly.
Okay, well, so we've got the pump. But like you said, the pump needs pipes.
Hmm.
How does blood actually move through this vascular system? Let's talk pressure, resistance, flow.
Right, the physics of the plumbing. Blood pressure, fundamentally, is the force blood exerts on vessel walls, usually measured relative to the outside environment.
Systolic, diastolic, highest and lowest.
Yep. And don't forget hydrostatic pressure. Gravity matters. In a standing person, the pressure in your leg arteries is higher than near your heart, just due to the weight of the blood column above it.
That's why they take blood pressure readings at heart level in the clinic.
Precisely, to get a standardized measure without that gravity effect messing things up.
But you mentioned earlier, it's not just simple pressure driving flow, it's this total fluid energy thing.
Exactly, this is key. Blood flows from higher total energy to lower total energy. And that total energy includes the pressure from the heart, yes, but also the kinetic energy, the energy of motion, and the potential energy due to height or gravity.
Okay. So that explains.
Why blood keeps flowing out of the ventricle into the aorta for a moment, even when ventricular pressure drops below aortic pressure. It's got momentum, kinetic energy carrying it forward.
Or why blood flows down into your legs when you stand, even though the simple arterial pressure in your legs is actually higher than in your aorta because of gravity.
Right, because the total fluid energy, factoring in that potential energy drop due to height, is still higher in the aorta. It's the total energy gradient that matters.
Okay, that clicks. So how do we predict flow rates? The Poiseuille equation?
The Poiseuille equation is central. It tells us flow rate depends on the pressure difference driving it, the fluid's viscosity, the length of the tube, and this is the kicker, the radius of the tube raised to the fourth power.
Or to the power of 4.
Yeah, which means tiny changes in vessel radius have a huge impact on flow. Halve the radius, flow drops to 1/16.
Wow. That's how the body controls blood flow so effectively then, by tweaking vessel diameter.
Absolutely, that's what arterioles do. And resistance to flow is inversely proportional to that radius to the fourth power. Small changes, massive effects.
And this flow isn't free, energetically speaking. There's friction, heat loss.
Right. As blood flows, especially in smooth laminar layers, there's internal friction because of viscosity. This converts the energy of motion, kinetic energy, into heat. So the pressure drop you measure across a blood vessel, that represents the energy cost of pushing blood through that resistance.
Arteries have low drops, low cost. Capillaries have big drops, high cost.
You got it.
Okay, let's focus on mammals and birds now. These high-performance closed systems, what really defines them?
They both independently evolved remarkably similar systems. Key features. Closed system, blood stays in vessels lined by endothelium. That lining itself is super active, by the way.
Secretes stuff, responds to hormones.
All sorts. And crucially, a series arrangement. Pulmonary circuit, lungs, then systemic circuit, body. All blood going to the body tissues is fully oxygenated first, maximizes O2 delivery for their high metabolisms.
And high pressure.
The highest systemic arterial pressures, needed to drive high flow rates against the high resistance created by billions of tiny capillaries.
Let's break down the vessels. Arteries, thick, muscular, elastic. Why the elasticity?
Super important. That elasticity damps down the pressure pulses from the heart pressure damping, and it stores energy during systole, releasing it during diastole to maintain pressure and keep blood flowing, the pressure reservoir effect. Smooths things out.
And Laplace's law explains why smaller arteries don't need walls as thick as the aorta, even with high pressure.
Exactly. Wall tension depends on both pressure and radius. Smaller radius means less tension for the same pressure, so the walls can be thinner but still strong enough.
Then we hit the microcirculation. Arterioles, capillaries, venules. Arterioles are the control gates.
Absolutely critical. They have thick, muscular walls capable of vasomotor control, constricting or dilating. By changing their tiny diameter, they act like taps, massively controlling blood flow into the downstream capillary beds.
And this is controlled by nerves, hormones, local signals.
All of the above. Think about blushing, or sending more blood to muscles during exercise, or less to your gut after a meal. That's arterioles adjusting flow. It's a key feature of closed systems' precise control. Nitric oxide is a major player here, causing vasodilation.
Then the capillaries.
Yeah.
The actual exchange sites.
The main event, Walls often just one cell thick, super thin, huge surface area, barely wide enough for red blood cells to squeeze through single file. This maximizes the efficiency of oxygen, nutrient, and waste exchange between blood and tissues.
Incredible density, too.
Right.
Meters of capillaries per tiny bit of muscle.
Astonishing. And the body can grow more if needed angiogenesis. After injury, during training, if oxygen is low.
And then veins bring it all back?
Yep. The low pressure, low resistance return journey,
Yeah.
thinner walls, they rely on one-way valves to prevent backflow, especially in the legs, and the skeletal muscle pump muscles contracting around them, squeezing blood towards the heart.
And they hold a lot of blood.
Yeah.
Capacitive.
Very much so. They can stretch easily, acting as a volume reservoir for the whole system.
Okay, fluid exchange in those capillaries. Starling Landis hypothesis, how does that work?
It's a balance of forces. Hydrostatic pressure, the blood pressure inside the capillary, pushes fluid out. Colloid osmotic pressure, due to proteins dissolved in the plasma that can't easily leave, pulls water in.
So out versus in.
Right. At the start, arterial end of the capillary, hydrostatic pressure is usually higher, so fluid filters out into the tissue. As blood moves along, pressure drops. At the end, venous end, the osmotic pressure is now stronger, pulling fluid back in.
Does it all get pulled back in?
Usually not quite. There's often a small net loss of fluid out into the tissues, and that's where the lymphatic system comes in. It collects this excess tissue fluid, now called lymph, and returns it to the bloodstream later, prevents swelling or edema.
The lymph system's role. And the lungs.
Mm.
The pulmonary circuit's different, designed to stay dry.
Critically different. It's a low-resistance, low-pressure circuit, much lower than the systemic side. This low capillary pressure in the lungs is vital because it largely prevents that net fluid filtration out.
Keeping the air spaces free of fluid for gas exchange.
Exactly. If fluid built up pulmonary edema, it would be life-threatening.
Okay, exercise. The system ramps up massively. How?
It's a coordinated effort. Oxygen delivery equals cardiac output times the oxygen difference between arteries and veins. So during exercise, you increase both.
Cardiac output goes way up.
4 to 7 times maybe, by boosting heart rate and stroke volume. And your muscles extract way more oxygen, so the venous blood comes back much lower in O2.
So you're pumping more blood and getting more oxygen out of each drop.
Precisely. And crucially, overall systemic vascular resistance drops significantly.
How?
Massive vasodilation widening of the arterioles going to your working muscles. This prevents your blood pressure from shooting dangerously high despite the huge increase in cardiac output.
And blood gets redirected.
Big time. Active muscles might get 70–80% of your total blood flow, up from maybe 20% at rest. Blood is shunted away from places like your intestines, kidneys, inactive muscles. Brain flow, though, stays pretty constant.
And training helps. More capillaries.
Endurance training increases capillary density in muscles, yes. Better plumbing for oxygen delivery.
Species differences are amazing. Giraffes always come up.
The classic example. Small mammals have way higher heart rates. Think 600 beats a minute for a mouse to get high weight-specific cardiac output. Birds tend to have bigger hearts, higher pressures than mammals of similar size.
But the giraffe, that neck.
Standing 5 meters tall, brain maybe 1.6 meters above the heart. It's a challenge. The consensus is they need extraordinarily high aortic pressure, maybe 220 millimeter Hg systolic, just to push blood up that column against gravity to perfuse the brain.
Incredible pressure.
Yeah.
And countercurrent exchangers. Clever heat trick.
Yeah, these are neat. Arteries and veins running right next to each other but in opposite directions allows very efficient heat transfer between them.
Useful.
Keeping testes cool in mammals, minimizing heat loss from legs or flippers in the cold, cooling the brain sometimes, keeping muscles warm in some active fish. Even crucial for inflating fish swim bladders. Lots of applications.
Okay, let's broaden out again. Fish circulation, the basic vertebrate plan.
Mostly closed systems, yes. But the key difference is the series layout. Heart, gills, body, heart.
So the heart pumps blood to the gills first.
Right. Then that oxygenated blood flows directly from the gills to the rest of the body without going back through the heart first.
So the heart's pressure has to push blood through both the gill resistance and the body resistance.
Exactly. That's a major constraint. And often their heart muscle itself gets oxygen from the deoxygenated blood flowing through its chambers, as we discussed with the spongy myocardium. Their overall pressures and outputs are generally lower than mammals' birds, matching lower metabolic rates.
And that myocardial oxygenation strategy really links to lifestyle in fish.
Strongly. Active fish, like tunas, have big hearts. Lots of compact myocardium with coronary supply, high outputs, high pressures. Less active fish have smaller hearts, mostly spongy muscle, lower outputs.
And the sockeye salmon example showed it's even plastic within a species.
Amazing plasticity based on migratory effort. And when fish exercise, say a trout, they boost cardiac output mostly by increasing stroke volume and pull more O2 from the blood. But that makes the blood in the heart even less oxygenated, which could potentially limit the spongy myocardium's performance, a potential bottleneck.
So what about air-breathing fish? Is their circulation set up a disadvantage? It seems like oxygenated blood mixes with deoxygenated blood.
That's the traditional view. They often have air-breathing organs, modified swim bladders, parts of the gut that empty oxygenated blood into systemic veins, so it mixes with used blood before going back to the heart.
Like the electric eel pumping only partially saturated blood. Seems inefficient.
That was the thought why recycle deoxygenated blood. But the newer perspective flips it. Maybe that mixing actually boosts the oxygen levels in the blood flowing through the heart chambers, which is good news for their spongy myocardium.
Ah, so it might be an adaptation for the heart muscle's sake, not a flaw in systemic delivery.
Exactly. It reframes it as potentially advantageous for their specific physiology.
That's a cool rethink. Lungfish, then, seem more specialized for separating the flows.
They do. They're thought to be close to the ancestors of land vertebrates, and they can achieve pretty good separation. Veins from their lungs connect directly to the left side of the atrium, and the heart itself has partial divisions.
And studies show the separation works.
Yeah, using tracers and oxygen sensors, they've shown oxygenated lung blood tends to go to the body arteries, while deoxygenated body blood tends to go to the lung arteries. Not perfect, but significant separation.
And this helps intermittent breathers.
Hugely. Because the heart isn't fully divided, they can redistribute blood flow. When they surface and fill their lungs with fresh air, they can send more blood to the lungs. When they dive and lung oxygen drops, they can reduce blood flow to the lungs and send more to the body. It's ventilation perfusion matching. Very clever.
Okay, moving on to amphibians and non-avian reptiles, masters of selective distribution, you call them.
Their systems are really fascinating, revolving around selectively distributing oxygenated versus deoxygenated blood, handling pressure differences, and redistributing flow, especially since many are intermittent breathers too.
Anatomy. 2 atria, but incompletely divided ventricle.
That's the key feature. 2 separate atria receiving blood from lungs, left, and body, right. But the ventricle isn't fully split. Often spongy myocardium inside, maybe a compact layer outside.
And even frogs, with no ventricular septum at all, can manage selective flow.
Remarkably well. Bullfrogs can direct most lung blood to the body and most body blood to the lung skin. How they do it so effectively without a physical wall is still debated. Complex fluid dynamics, maybe?
And skin breathing complicates things.
Well, oxygenated blood from the skin mixes with the systemic venous return, again, traditionally seen as inefficient mixing, but now also considered in light of potentially helping oxygenate the heart muscle.
Okay, non-avian reptiles, turtles, snakes, lizards, also incomplete ventricles.
Usually partly divided into 3 chambers by muscular ridges. They also achieve dramatic selective distribution. Some, like pythons and monitor lizards, can even create temporary functional separation during contraction, allowing them to have high pressure in the systemic circuit, but keep low pressure in the pulmonary circuit, like mammals.
Protecting the lungs.
Exactly. Others, without that ability, have to keep pressure low in both circuits. And like lungfish, they are masters of redistribution work. Diving turtles might send hardly any blood to their lungs during a long dive.
And pythons remodeling their hearts after eating.
Incredible example of physiological remodeling. Massive increase in heart mass and contractile proteins driven by gene expression changes after a huge meal. Shows extreme adaptability.
Crocodilians are different again. Complete ventricle split, but shunting.
They are unique. They have a fully divided 4-chambered ventricle, like us, but they have 2 systemic aortas leaving the heart and a connection between them called the foramen of Panizza.
Okay, what does that allow?
During normal breathing, they function much like us, with near-perfect separation. But during diving or breath holding, they can use valves and pressure changes to deliberately shunt blood away from the lungs, a right-to-left shunt, sending more deoxygenated blood into the systemic circulation. Again, adaptive for intermittent breathing.
So the big picture for vertebrates. It's not a straight line to mammals, birds being best.
Absolutely not. That's a crucial takeaway. These other vertebrate plans aren't less evolved. They have unique advantages perfectly suited to their lifestyles, especially intermittent breathing. An incompletely divided heart can actually be more flexible and efficient for those strategies.
Fascinating. Let's briefly touch on invertebrates. Some have closed systems too, like cephalopods.
Right, squids and octopuses. Active predators need efficient circulation. They have a main systemic heart, plus two auxiliary branchial hearts just before the gills to boost pressure there. Gills and body are in series.
Similar pressures to fish.
Reasonably high pressures and flow rates, yeah. But they have a big problem, physiologically speaking.
What's that?
Their blood pigment, hemocyanin, carries much less oxygen than our hemoglobin, and it makes the blood quite viscous. Plus, they use almost all the oxygen in their blood, even at rest, no venous reserve.
So to get more oxygen during activity,
They have to pump blood much, much faster. They can't just extract more oxygen from the blood like we can. The entire burden falls on dramatically increasing cardiac work. This might be a key reason why fish ultimately outcompeted them evolutionarily in many niches.
that's a huge limitation. Okay, finally, the open circulatory systems. What defines them?
Here, blood, usually called hemolymphs, leaves distinct vessels and flows through open spaces called lacunae and larger channels called sinuses, directly bathing the tissues. There's no strict separation between blood and the general extracellular fluid.
So blood just washes over the cells.
Pretty much. Decapod crustaceans, crabs, lobsters are a good example. Single-chambered neurogenic heart pumps hemolymph out through arteries.
Which aren't muscular.
Mostly not. Control happens at the start of the arteries via these little muscular valves called cardioarterial valves. Then the hemolymph flows through lacunae in the tissues, drains into sinuses, goes through the gills for oxygenation, and back to the heart via the pericardial sinus.
And unlike fish, blood goes through the body first, then the gills.
Yes, that's a key difference in layout.
Now, the surprising thing is that open systems, despite low pressure, aren't necessarily slow.
That's the paradox. They operate at low pressures, maybe 1-2 kPa in a lobster, but they can achieve very high flow rates, sometimes circulating their blood volume much faster than fish do.
How? If pressure is low.
Because the resistance to flow is very low. Flowing through wide-open sinuses is much easier than squeezing through tiny capillaries. They need that high flow because their hemolymph usually carries less oxygen.
So low pressure, low resistance, high flow.
Yeah.
Different strategy.
Exactly. And we're learning they have more sophisticated control over flow distribution than we used to think, using those cardioarterial valves and maybe other mechanisms.
And the ultimate outlier. Insects, super active, high oxygen needs,
Yeah.
but a pretty basic open system.
The insect paradox. High metabolism, yet a simple dorsal tube heart, no capillaries to speak of.
The tracheal system.
Bingo. Their breathing system is a network of air tubes, tracheae, that delivers oxygen gas directly to the cells, completely bypassing the circulatory system for O2 transport.
So the circulation doesn't need to be high performance for oxygen?
Nope. It can focus on slower tasks like moving nutrients, hormones, waste. It's a key lesson. Circulation can stay simple even in active animals if it's unburdened from oxygen transport.
What a fantastic journey through animal circulation, from our own hearts to lobsters, lungfish. Every system really is this masterpiece of adaptation.
It really is. And what really strikes me is how systems we might have once called simple or less advanced, like in air-breathing fish or those open systems, they're actually incredibly finely tuned with their own unique advantages for their specific way of life.
It makes you wonder, doesn't it? What else in biology are we maybe looking at through, human-centric or overly linear glasses?
Exactly. Could these different strategies inspire new engineering designs, challenge our ideas of what's optimal? What might we learn from animals we haven't studied as closely, or just by asking new questions about old assumptions?
Lots to think about. It encourages you to keep questioning, keep exploring these hidden biological marvels. A huge thank you to everyone listening for joining us on this deep dive. We hope you enjoyed it.
Thanks for tuning in to the Last Minute Lecture family.